Hey guys, sorry I'm running a little bit late. Let me pull up what we're going to be doing today. Wait just a couple minutes to make sure everybody is logged in and able to get onto Zoom. I'm assuming we'll probably get a couple stragglers. So we'll get started here in just a couple minutes. We're going to play through the Kahoot, but like I said, I'm going to spend quite a bit of time on explaining the different aspects of things. It's in the chat. 35592. Yes. So before we get started with the review here for the Kahoot, I am really quick going to remind you guys of some of the study resources that we have going on Canvas already. So if you go to Canvas and go into the modules, this is week 5. It'll go. Here it goes. Let it catch up. Here we go. Okay, so here in week 5, we can see we have the lab manual here, which will have all of the information you could possibly get tested over. But if you want to break it down into learning objectives, we do have the lab learning objectives here set up in a similar fashion. We have the general ones up at the top that apply to every lesson, and then lesson specific ones. And so our lab midterm is going to be over lab exercises 1 through 11, right? So we did 11 yesterday, 1 through 11. And so you want to be sure that you get all those learning objectives. Plus you want to be covered for anything related to lab safety as well. So be aware of that too. And so we have that. That's one way you can break down your studying. We have the lab midterm study sheets. And these are those sheets that are at the end of each lab exercise. Again, we've got lab safety on there. This is any of the stuff that I could test you over on that stuff. We have aseptic technique, we have colony morphology, and each one has basically a little sheet that's gone along with that exercise with information of what is going on for each thing. So definitely for each stain, know what your primary stain is, what your if there's any mordants, if there's any decolorizers, if there's counter stains or secondary stains, and stuff like that. So we'll kind of go over a little more detail in that with this um, review. But the information that you would be using to follow along with the review, it should be in these study sheets and it should be covered in your learning objectives for the lab. Okay. I don't remember what this is. Right. So this is just a very basic rundown of each of the lab exercises that we've gone over, major concepts from each one. Not everything, right? But the major concepts from each one to help you organize your information and understanding. Then we have the transformation summary, which should be just a breakdown of P-Glow, what it is, and what products are made on what media. So more watered down version of that sheet that came along with the transformation. All right, and we don't need the lab project right now, but if you scroll down to the bottom where there's additional resources, prep guide for lab exams, it basically is walking you through like how to look at your lab exercises and break down what you need to know from that. If you want to go from the study sheets, that's great, but the lab exercises just have more information and a little bit more spelled out. And so this kind of walks through that. Just basic rundown of that stuff. Waiting for my page to catch up with me. Then we have two different study guides. These are not required, they're for study purposes only. Study guide 1 is basically just questions about the concept or like having you list stuff or answer a question about the concepts. But study guide 2 is having you create charts or more interactive aids for studying. So it'll have things like make a chart, draw depictions, draw stuff like this. And so this is a little bit just more interactive chart development and stuff like that. So those are all there to help you guys prepare for the lab midterm. There's plenty of resources on here, just many, if not more, than what we usually get for our unit exams. So please remember to use those. Don't just, I mean, I'd say you could just rely on the lab learning objectives to get you through the lab stuff. But if you're looking for more stuff to test your comprehension or application of those concepts, then go through week 5, which is the week that we're on. We only have weeks 5, 6, 7, and 8 left, and we're about to knock through week 5 pretty quickly. So be aware that we're almost there. You don't have that much longer left of the course. But the study resources are here for you to prepare for the lab stuff. So we're going to make sure you're logged into the Kahoot again real quick. Some of us might have got kicked out or something like that, or just joined and not joined on the Kahoot, but we're going to be doing the Kahoot for our review. And again, I'm going to go through all of the questions with it. Let's see. All right, so we're going to go ahead and get started. All right, so review your lab safety stuff. Again, if it's on that study sheet, you're for sure expected to know it for the exam. Part of the way that I test your lab safety understanding is just that you are expected to show up with your lab coat and your closed-toed shoes, and that you can lose points if you don't have one or the other, which you guys got your notification about that. The email that was sent out to remind you guys. I'll just bring that up really quickly. Bring your laptops and your tablets. This is a Canvas-based exam, just like your unit exams, but it's all going to be multiple choice. There's no short answers on this one. Wear your lab coat and closed-toed shoes. Otherwise, you could be missing points on your exam if I have to loan you a lab coat or if you don't have closed-toed shoes. And if you don't have closed-toed shoes or if everybody borrowed a lab coat and I don't have any extras left, then you're going to be asked to go to the testing center to take your test. And whatever grade you get on the testing center, you're going to have 5 points removed. You do want to keep your phones with you in your lab coat pocket, just in case you do need a dual push or I have to like reset it again. It happens every exam that we have. Somebody gets locked out of their exam and I'll have to reset it and you might need a dual push. So please keep your phones in your pocket on silent. Do not disturb it. Turn off your notifications. I had a student who like, oh, I have an alarm that goes off every day at noon, so my alarm's going to go off. No, turn it off for the exam. So keep that in mind. Be respectful of the other people there. It's not that much of an inconvenience for you to make a note to yourself to turn your alarm back on later. So please keep your phones on silent and in your pocket in case you need it for a duo push. I'm not going to allow you to go back into your bag to go find your phone to do a dual push. If you don't have your phone on you and you need a dual push, I'm going to give you a paper exam and you're going to have to start over again. You may only have your phones out if you have like told me, oh, I'm locked out. Will you come reset it for me? I'll come reset it for you. And then if you need to do a push, I'll be there for you. But your phones shouldn't be out at any other point outside of that. I'm not as picky about Apple Watches or smart watches just because I am walking around the whole time with this exam. The odds of you having the time to actually search for a thing on your watch without me noticing is going to be, you're going to be hard pressed to achieve that successfully. And so I'm not as picky about that. Yeah, so definitely more concerned about your phones themselves. Yeah, so only have that out if you need it and with my, supervision and stuff like that. If I did catch somebody on their phone without getting my approval to have their phone out, you will automatically receive a 0 on your exam. Okay, I feel like that's pretty straightforward. Let's go ahead and do the thing. All right, where do we dispose of broken glass? Easy question to start with. Right. So it's pretty straightforward. This is going to be the sharps container. If I were to ask you guys where would we put our slides when we were done with them, they also go in the sharps container, right? Because they have edges that could poke through the bio trash. So even used slides, even when they're not broken, will go in the sharps container. So remember that. We discussed that in the beginning of the lab course. Okay. All right. So the meat of it. This is going to be our stains. You'll need to know each of the steps and what order those steps come in. Each stain that is used for the procedure. My hair is just not cooperating today. It's really bothering me. Each stain that is used and its purpose in that staining procedure. We don't ever have superfluous extra steps in any of this stuff. So what is the purpose of each of the steps that we are doing for each stain? And they all have a purpose. Sometimes it's as simple as, oh, well, when we decolorized, now negative cells will not have a color. And so we want to be able to see them with contrast. And so that's why we have counter stains. And so it could be as simple as that. Know what a positive and a negative look like and identify each in a mixed sample. So on the questions, when it is relevant and when I feel like you need to know that it's a pure culture versus a mixed culture, that will say that in the question. So I might say this is a mixed sample. That means that there's more than one species of bacteria in there. And so if it's a mixed sample and I'll ask questions about that relating to whatever your slide is that you're looking at. Um, so know what a positive would look like for each stain procedure and know what a negative would look like for each stain procedure. So something else that I want to bring up, what a positive result looks like for a gram stain and what a negative result looks like for a gram stain is a completely different concept than what a positive stain and what a negative stain is. So if a stain has a property itself, like let's say crystal violet, for example, crystal violet itself is, well, I don't know, I would ask you guys, do you think it's a positive stain or do you think it is a negative stain? It's a positive stain. It doesn't matter if your result of your gram stain is red or purple, but crystal violet itself as a stain mixture is a positive stain. And why is that? Because it stains the cell surfaces, has a positive charge that's associated with the negative molecules on the outside of the cell. And so it is going to associate with the outside of the cell because of those charges. And so that has to do with the charges of the stain and the charges of the cell that you are using, positive and negative stains versus a positive and negative result of a staining procedure. So those are different things. And what happens if you skip a step in one of our differential stains? Be aware of what would happen if we forgot a step. So what is the purpose of heat fixing? Select all that apply. You won't have select all that apply on my test. It'll be one answer will be correct, but there will be all of the above or none of the above. Those will be choices on some of the questions. Right. So this is going to be adhering it to the slide and killing the microbes. Nobody chose these two over here, but some people did say stain it. Is the heat fix itself staining the slide? No, it might be preparing it and making it better suited for the staining procedure itself, but the heat fixing isn't actually part of the stain. It's like it's not staining the cells, right? So that's what the question is asking, and be sure that you're reading and understanding the answers in the context of the question. There are some questions on this test, like I've said in the class, there's not going to be, clinical situation where you're going to have to do a lot of critical thinking. I don't feel like there's very many questions on our exam that are like actual critical thinking questions, but your choices will have to be made with critical thinking, just using your brain. Because some of the answers, it's like, what is the best answer for the question? So be aware of that as well. What is the best answer? And so read through your choices and make sure that you're understanding all of your choices too. What is the counter stain used in the gram stain? Alrighty, so this is going to be saffronin. So the steps of the gram stain are your first step is going to be crystal violet. That's your primary stain. And like pretty much like your positive or negative result depends on how crystal violet behaves with the cell, right? So your crystal violet is your primary because of that. It also happens to work out that all of our primary stains are the first stains that you use. So there's that too. So primary stain is going to be crystal violet. And then we have two steps after that, which we're going to get to, I think, in a couple of questions. The decolorizer step and the mordant step, and then the decolorizer step. And then the counter stain is the last step. And so it's almost always going to be the counter stain is the last step. And so think about like, okay, if the last step, what are we doing? Oh, the last step is to give color to our negative cells. What are we using to do that in our gram stain? That's the saffronin. All right, what color will a gram-negative bacteria be after the gram stain? All right, so gram-negative bacteria will be red. This is because of the crystal violet should get stuck in gram-positive's cell wall. And so remember that the gram stain, what it's really doing is differentiating cells based on their cell envelope. So the cell envelope of a cell has to deal with your inner membrane, which we call the cytoplasmic or cell membrane or plasma membrane. Those are all just different words for the same thing. So that cytoplasmic membrane, on the outside of the cytoplasmic membrane, you have a wall, typically, and then the outside may have an outer membrane if you're gram negative, right? Depending on how thick that wall is, might trap your crystal violet in there. And if you have a thick wall like our gram positives have, you will trap the crystal violet in there and you will hold on to that purple color of the crystal violet, even whenever you rinse it with our decolorizer, which in the gram stain, our decolorizer is ethyl alcohol. And that removes any crystal violet that didn't get locked in place by our mordant, which was iodine. And that only gets locked in place because you have a thick wall to like create that meshwork for that locking in place to happen. So gram negatives will get decolorized by the ethyl alcohol. And then when we counter stain them, if we're doing the stain properly, when you counter stain it, it should be red. Yeah, let's go on to the next thing. What is the first stain used in the capsule stain? Okay, so this is where I'll point out to you guys that we really only have, if we were to write out all the different staining procedures that we use, we really only have four. We only have four staining procedures. So definitely have a way in your mind for quick access in your mind to recall what the four stains are and like what a positive and negative would look like. If you can remember what a positive looks like for each of those four major stain types that we did, then that's going to give you a pretty good baseline of what how to answer the questions, really honestly. Like, yes, study the other details too, but this is what I'm saying is this. The different stain types that we have, the different differential stains that we did. Just the differential stains. Gram stain, acid fast stain, capsule stain, endospore stain. What other differential stains did we do? That's it. So I get a lot of my students during the lab midterm who will be like, it's a lot of colors and a lot of stuff to know. It's four differential stains. And so if you can remember gram stain, positive is purple, negative is red. What was the second one? Acid fast stain. So I'm going to make little notes down here as I'm going through this just really quick because this will help me keep my thoughts in order because otherwise I'm going to start talking off on one thing and not remember to come back to it. Of course, this is the day where I've literally already misplaced all of my pens ahead of time. All right. So gram stain, positive purple, negative red. That's all I'm going to write down for now. Acid fast stain was our second one. Acid fast. Positive was fuchsia, magenta, pink, whatever you want to call it. So we'll just put pink, bright pink. Negative for the acid fast stain was blue. All right, third one, the capsule stain, the one that this question was about. Capsule. Positive had a halo around the cells, right? A clear, colorless halo around the cells. That was what the actual capsule was depicted as. Now, that should say, if you have a halo around the cells, how do you get a halo? You have a background. That's important to remember. That is the most important thing to remember about the capsule stain. You're determining whether it has a halo around it, has a capsule around it, based on the background versus the cell itself. So this one isn't even about the color at all. So this one's just whether you have a halo or not, a colorless halo. The colorless halo happens because the capsule is a sugar-based, basically uncharged, sticky coating that it has, and it doesn't stain. And so the halo represents the unstained capsule. You'll have a background and you'll have the cell that's stained. You have to have that in order to see a colorless halo, right? So capsule, halo is positive. Negative for the capsule stain is no halo. The defining feature that allows you to see the halo is that there will be a background. Whether you're positive or negative for a capsule, you must have a background to know if there's a halo or not, right? All right, so the primary stain that we used for our capsule stain was Congo red. And Congo red stained our background. It basically gave us a background. So Congo red is a particular type of stain. When we mentioned earlier about the positive stains and the negative stains, and I told you positive stains are positively charged and then therefore are attracted to the negative charge of cells, opposites attract. So if positive stains stain the cell, what do negative stains do? They stain the background. Okay, so what kind of stain is Congo red? It's a negative stain. Okay. Whether or not you have a halo or not would determine whether you are positive or negative for the stain itself. Like this is where I come back to that. So I just said to you guys, you got to know the difference between positive and negative stains themselves, but also positive and negative results. Those are different things. Okay, so Congo red is our primary stain. This is what we mix our sample up with in the liquid and then spread it out, and then we let it air dry, right? Okay, just want to set us up for that before we move on. So I've got my little list here I don't have a way to like show you on my actual screen that we're showing here. We could annotate it, but I don't have a pen to write with, so it is just my mouse, so I can't. It would be it would be horrible if I tried to write on this. Just briefly, this is what I have so far. I've got my gram stain, what positive and negative look like, acid stain, what positive and negative looks like, capsule stain, what positive looks like has a halo versus negative looks like versus no halo. And I put background on here to remind me that's how you know if you have a halo or not. Okay. All right. And then our last one, our fourth staining procedure was our endospore stain with this thing. Okay. Fourth was endospore. Positive endospore. Don't just put green. Don't just put green. Don't. Okay. So a positive endospore stain, you will have what we could say organized structures that will stain green inside. Organized structures. They're endospores is what the structures are, right? And those organized green stained structures will be amongst a sea of red. And those red ones that would be red in the positive stain would be the vegetative or the sporangiums that are forming the endospores. But I'll just put red cells. So oftentimes, I would say the number one most missed question on my exams by far is people recognizing the endospore stain. Even when I tell you it's a pure culture, and then when people look in the microscope at my pure culture, and there's a ton of red bacilli, which are what shape? Rod shaped, right? So there's a ton of rod shaped cells that are red, and these weird bubble structures that have a tiny bit of greenish tinge on the inside. If that's a pure culture, cells don't have rod and circle shapes. If it's a pure culture means it's one bacterial species, and I shouldn't have to remind you of that. So if it's a pure culture of one bacterial species and you're seeing a bunch of rods and a bunch of clearly defined little circles, even if you can barely tell that they're greenish inside, that should be a hint to you that there are endospores in that sample. And so if you can't really see the greenish there, but you see that the reds are there, you can use the fine adjustment knobs on your microscope to see if you can catch that green in those little circles. And if you do, then you know that this is the endospore stain. Yeah, I do expect you to do that. Number one most missed question by far is that one. And I'm not dropping it. So I feel like if you only got 4 differential stains that you need to know, and one of them is the endospore stain, and you can see clearly if and I'm telling you that it is a pure culture and telling you that it's not a pleomorphic bacteria. It can't change its shape. Even a pleomorphic bacteria, you're not going to have rods and circles. You're going to have all these different shapes all over the place. And they're not going to be very clearly defined either way. So if I'm telling you it's a pure culture and I'm telling you it's not pleomorphic and you see rods and circles in there, that should tell you already that this is an endospore forming bacteria. And if you looked at the slides, that I provided, that I told everybody to look at everybody's slides, at everybody's tables, so you can see the different, like what it looks like. We had 2 tables with endospores. One of them had the little bubbles, and one of them had clear, like pill-shaped guys. But they're very clearly like a different aspect of what's going on based on the regular cells around them, the red cells around them. You have to be able to recognize that. So that's one you might want to put a little more detail for what a positive and what a negative looks like. So for endospore, I have organized structures that have greenish tint to them that are surrounded by red cells. For negative endospore test, I'm putting that only red cells. It should only be just red cells all over the place. There shouldn't be any defined structures all over around them. Only red cells if it's negative for the endospore stain. Okay? So this should hint to you that I have a tendency to put positive endospore on the exam. I just expect you to notice that this is the endospore stain. All right. So that's my little mini notes there just what that is. And so our first stain used in the capsule stain, if the capsule stain depends on a background being there in order for us to see our halo, and whether or not you have a halo or not is going to determine whether you're positive or not. In order to determine if a halo is there, you need a background. And the stain that we use for that is our negative stain. It is the only negative stain that we use in the whole lab is Congo red. All right, so we're getting our thoughts organized. We've got a little mini cheat sheet to work from. What type of stain is Congo red? Select all that apply. All right, excellent. Good job, everybody. It's a negative stain, and negative stains stain the background. Excellent. I'm glad you're listening now. Hopefully that sticks when you get to the exam, right? All right. This is a pure culture. Which stain is pictured? Pure culture means it's one species of bacteria. And on the exam, if it's relevant, I will also state that it is not pleomorphic. So you've heard me say that word pleomorphic a couple times. So that's one that you should probably be aware of what it means. Pleomorphic means it has basically no defined shape. It can have a whole bunch of different shapes. Pleo means many, morph means shape, right? So this was a pure culture. We saw that there were red cells. Sometimes there is it. We see that there's red cells here, and we see there's these weird bubble shapes all throughout. This is a pure culture. Why would you have circle shapes and rods in a pure culture? Well, the circle shapes are the endospores. That's why. And so what color are these? It might look bluish to you. It might look greenish to you. It might look teal to you. I don't care. But what stain results in us being able to differentiate between the cells and these other structures? This is the endospore stain. There's only 4 stains you got to know. You need to know the details about them, but there's really only 4 differential stains. All right, what is the term for the red structures in the image? Alrighty, so the red structures are the vegetative cells. Those circles that we saw when we look at the media, the circles, these guys here, these circles, these are the actual endospores. So we see some of these guys are like rods, and then there's like a green thing at the end of the rods. That is this endospore that's being formed. So that structure where you have a rod with a green thing at the end, that's the sporangium. That's the vegetative cell that's forming an endospore. But the red structures are all just vegetative cells, the active multiplying version of those. All right, which of the following bacteria would be acid-fast positive? So this one is the one that gets a lot of people tripped up. We know myco. We usually can remember myco. We know that. But in the acid fast stain, we are looking for an acid, technically, quote unquote, but it is a wax. So just like we have fatty acids, this is a type of wax. It's called mycolic acid and is a wax that might be on the surface of certain gram-positive organisms. And so the only genus that you need to know for my course that's able to have mycolic acid wax on its surface is Mycobacterium. And so my silly way of remembering that is there's 2 mycos that you'll come across in my course, mycoplasma and mycobacterium. And so mycoplasma has the word plasma in it. And so that's why I remember if you're carrying around the cup of plasma and you spill the cup of plasma, like somebody's blood, right? Plasma, like I mean plasma. So if you're carrying around a cup of someone's plasma, they just donated at CSL or something like that, and you've got their plasma and you spill it, does it maintain its shape? No, it's like it's liquid and it goes everywhere because mycoplasmas are pleomorphic. They're the only ones that I'd ever tell you that could be pleomorphic. and they're pleomorphic because they don't have a cell wall at all. So they're technically gram negative because they have two membranes, but they don't have any wall at all. That's the mycoplasmas. The mycobacterium doesn't have plasma to skew you away. It's a bacteria with myco. Why does it have mycobacteria? Because the myco is the mycolic acid. And so that one is the one that we would pick for this. This is a stupid way of me remembering it, but I do remember it that way. You can remember mycoplasma and mycobacterium. The only hint here, it's a bacteria, cool, with myco. If you look at mycoplasma, there's kind of myco and plasma. What's plasma? Oh, the cup that you spill, it doesn't have a shape. Okay, this is the one without the cell wall. Like silly, but it works for me. Maybe it'll work for you. I don't know. But this is mycobacterium, anything mycobacterium. And so two very medically relevant. There's more than just one that can cause disease, more than just two that can cause disease. There's several that can cause disease. But the two biggest mycobacterium species that can cause disease are going to be Mycobacterium tuberculosis and Mycobacterium leprae, which is leprosy, cause of leprosy. So Mycobacterium is the one that has mycolic acid. If it has mycolic acid, then the primary stain that we use, which I think is one of the questions on here, if it's not, we'll come back to it. But if it's acid-fast positive, then we already know from our little cheat sheet notes, if it's acid-fast positive, it would be pink, that fuchsia color. There's a hint there, but it'll be pink, right? And so why is it staining with our primary stain that makes it fuchsia? Because it holds fast, because the wax absorbs our lipid-soluble primary stain and holds fast even when we rinse it with hydrochloric acid and alcohol mix, which is our acid alcohol. All right, what is the primary stain that we use in the acid-fast stain? We're paying attention. You saw that my notes said that our acid-fast, I put pink, but I said we could call that fuchsia or magenta or something like that. Well, there's a little hint in one of those. All righty, carbofuscin. Good job, guys. So carbofuscin stains at fuchsia, and it'll always be called carbofuscin. So I feel like that hints a little pretty good at what the positive is going to look like. And carbofuscin is lipid soluble, so that's why it stains that wax. All right, so in which staining procedure did we use steam? Yes, this is going to be the endospore stain. So we actually used steam or heat to force our primary stain, which was, I'm sure it's going to be a question on here, but I'm going to go ahead and remind you. The primary stain for the endospore stain is malachite green. And so that green, it's a greenish, tealish, bluish color, but we're forcing that greenish color into the endospores, which have that thick cortex coating. The steam helps force that in there. And then whenever you rinse it off with water, the vegetative cells aren't going to hold on to that color. But because our endospores have that thick cortex, the water doesn't rinse out the green. And so it keeps holding on to it. So our decolorizer in the endospore stain is actually just water. It's just deionized water. But steam acts as our mordant in our endospore stain. It's a mordant because it's helping fix it into place where it needs to be. So that would be steam. It's the endospore stain. Next question, which of the following structures allow for bacterial motility? Alrighty, so this is just the flagella. Flagella are what bacteria use, and they actually use a little motor to spin it. And we talked about the different arrangements of the flagella around the cell as well. Remember, this was also in our lecture exam, Unit 1. So the peritrichus is where they're all over. The monotrichus is where you just have one flagellum. That's the singular for flagella is multiple. Lophotrichus is having a tuft at the end. And we have also amphitrichus, and amphi always means both. So flagella had each end is amphitrichus. Yes, so cilia, just to kind of go over these other choices, bacteria don't have cilia. This is a unique characteristic of eukaryotic cells. Some eukaryotic cells have cilia. Fimbriae, these are for attachment typically. And pili, these are those sex pili that we use to transmit information through the channel from cell to cell. It's a way of horizontal gene transfer. So our correct answer here is flagella. All right, so that's it for our review of the stains part. Remember what a simple stain is. A simple stain is usually just going to use one positive stain. So I mean positively charged. It'll stain the cells themselves, as opposed to negative stains, which stain the background, right? So simple stain usually just uses a simple one positive stain. We used methylene blue in ours, but it just gives contrast. That's the only thing. It's not going to tell you the difference between a positive and a negative result. Those are our differential stains. All the differential stains that we did were gram stains, acid-fast stain, capsule stain, and the endospore stain. The wet mount is the procedure that we did to look at whether our cells were motile or not. So we looked for them swimming and all that, right? And we talked about the difference between Brownian motion versus actual true propulsion by our motility from our flagella, right? So if you were able to swim across your microscope view, then you had to have flagella. But if you're just sitting there vibrating, you're probably just getting moved by the actual water in the slide causes the cells nearby to vibrate. That's Brownian motion. So that's not true motility. And so that's not actually really a stain, but it is something that we used to differentiate for whether you have motility or not. So it is a differential slide preparation, but it's not really a stain, right? So our main four differential stains were the gram stain. It differentiates based on that cell envelope and really more specifically the cell wall, whether you have a thick cell wall or a thin one. And remember, in bacteria, that cell wall is made up of peptidoglycan, right? The acid-fast stain just tells you the difference between bacteria that have or don't have mycolic acid wax on the surface because carbofuscin, our primary stain, is lipid soluble. So it actually binds to the wax and will stay stuck to it even when we rinse with acid. The capsule stain is just checking to see if you have a capsule or not. And most of our bacteria, there might have been one or two that we weren't sure about and we might have put didn't have a capsule, but most bacteria are going to have capsules. They're pretty useful structures, right? So we're talking about sugar coatings that are densely and tightly associated with the cell. So you get a nice defined halo around them. These are useful for attaching to surfaces, attaching to tissues inside of a host, as well as for evading phagocytosis by white blood cells in the host. And also for creating biofilms. So those are all things that capsules can help aid in. And then the stain, when staining for the capsule, we're staining the background and the cells. A little more detail on that capsule stain procedure. We used Congo red to create the actual background because it's a negative stain and it's repelled by the cell surfaces. So we create a background with that. And then Menneval's stain is a secondary stain on that one. Menneval's stain just changes everything to a different, like a pinker color of itself. So some of us, it turned it kind of a bluish color. Some of us, it turned it a pinker color, some to a little purple. And it doesn't really matter as long as you can see the cell and the background. If there's a background on your slide, it's the capsule stain. None of the other ones have a background. None of them. So if it has a background, it's the capsule stain. The stain that made the background was the Congo red. The Menneval's is just there to help stain the cells. So you can now tell the difference between the cell and the background and then see if there's a halo or not based on whatever happens with your primary and your secondary stain. Now we didn't heat fix with the capsule stain, right? Why was that? Now, if we think back to the capsule stain, we literally just mixed it with that Congo red, spread it across our slide, let it air dry, added Menneval's on there, then we waited again, and then we rinsed that off in our Copland jars. We had to rinse it off in the Copland jar because we didn't ever heat fix. And so we don't heat fix on purpose with this one. And that's because if you take your Congo red, mix it in, spread it out, and air dry it, if you were to heat fix it after that, it would kill your cells and help them adhere to the slide, yes, but it also shrinks them and it creates a fake halo. And so now you can't tell if there's a halo from the capsule or a halo from your cells shrinking. And so that's the importance of not heat fixing with the capsule stain. The wet mount, just whether it moved or not. We didn't use any stains in order to help us see the cells. We had to turn our light so that we could have contrast just based on the light differences there. And we looked for movement in our microscopes. If you moved, you have flagella. If you were just sitting there vibrating, that was Brownian motion. That's not true motility. And the endospore stain, this was one where we create our normal smear, we heat fixed it, then we added our malachite green to it. So something I want to be very clear about here. What was forcing the malachite green into the endospores was the steam. The only reason that we had the paper towel on there was to help keep the stain localized over um, our smear that we heat fixed. So the paper towel was just there to keep the stain there so we could keep it moist for the 10 minutes that was over the steam so it didn't dry out. It helps with keeping it from drying out and it also helps keep the stain on the actual slide. I mean, we saw what happened when that stain didn't stay on the slide. So it was meant to help with that. So the paper towel doesn't actually have function in the staining process. What has function is the steam and the stain. The paper towel is just kind of a helper with it. So paper towel is not a mordant. The paper towel didn't actually actively participate in the stain, and the stain works without the paper towel. It's just harder to get it to work properly. So that's why I want to bring up paper towel was not important in that staining procedure. It was just a helpful tool. But we stained with the malachite green. We put the steam under it for 10 minutes to help force the malachite green into the endospores if there were any. Then we rinsed it off well with water, and that should decolorize any vegetative cells. And then we will counter stain with safranin on that one, just so we can see the difference between that teal blue and the red. If you were red, you were vegetative. If you had that green tint on the inside, then you were an endospore. And that's generally how that stain worked. All right, before we move on to the next part, does anybody have any questions for me on the stains? Okay, we're going to move on. Next is the ELISA. This is, okay, well, it's going to ask you some questions, but I don't want to lead too much into it. But we did this yesterday. So this one should be pretty fresh in our memories. All right, so this is an immunologically based test. It's going to, I think, ask you what the E stands for or something on the next question, and that's fine. But it's an, enzyme-linked immunosorbent assay. Immuno. Any of our immunology-based tests, any of them, whether it is enzyme-linked immunosorbent assay, or if it is immunofluorescence, or anything with immuno in it, and most serology-based tests, those are going to be based on immunology interactions. And so that's always going to be antibody dependent. So either you're looking for the patient having the antibody using a known antigen, or you are looking to see if the patient has the antigen looking for a known antibody. So this is a very specific one where we got into detail on the specific one, but all immunologically based tests work on that principle of antibody-antigen interactions. We'll learn more about those in chapter 18 when you guys get to it, but I want to set you up for success with that the antibody-antigen interaction doesn't just apply to ELISA. It applies to a lot of immunologically based tests. All right, first question, what does the E in ELISA stand for? And I don't care if you call it ELISA or ELISA. I don't care how you pronounce it, as long as you know it's ELISA and you know what each of those letters stands for. Enzyme. Again, enzyme-linked immunosorbent assay. It's enzyme-linked because one of the antibodies that we'll use at the end, regardless of if you are direct ELISA or indirect ELISA, you're going to use an antibody at the end that is linked to an enzyme that's going to cleave a substrate, right? So that's why it's enzyme-linked. Immuno means it's dealing with antibody antigen. Sorbent because adsorbent. So ADSORBENT. A-N-T. I'm not gonna make you write it. So that adsorbent, it's E-N-T. That aspect has to deal with the fact that these things are sticking together. So we're either sticking antibody to antigen or we're sticking the antibody to the well. It's a whole bunch of things just sticking on to things, okay? So that's the sorbent part. And it's immunosorbent because it's depending on antibody antigen to cause things to stick together. Assay. I don't think that needs more explanation. All right, next. What is an indirect ELISA looking for? The question that we're asking when we do an indirect ELISA. All right. Does the patient have the antibody? Exactly. So if we were asking what the direct ELISA is looking for, that would be, does the patient have the antigen? It's always in immunology, if we're doing an immunological based test, antigen or antibody. And they're all either indirect or direct. Again, it doesn't just apply to ELISA. It applies to immunofluorescence. It applies to Western blots. It applies to a lot of things. So antigen or antibody, if you're looking for the antibody, that's an indirect way of seeing if this person has come into contact with the disease. But if you're looking for the antigen, that's directly looking for the disease. Beautiful. All right. Know your principle of how the assay works. You definitely want to understand an antibody, what it is, and what an antigen is. Indirect versus direct, we kind of just went over that. How the color change happens, the whole reason that you go from non-colored to the purple. It said green to purple. Some of you guys might have seen a greenish tinge. Most of you guys probably saw no color there. It was the same thing. You're looking for whether or not it turns purple, right? And so if that color change happens, it's positive, and that's how it always is. So you don't have to worry about like, oh, this time it turned the color change. Was that positive or negative? Every time in these immune-based tests, if it changes a color, it's positive. And that's because the enzyme was there, because that enzyme that was linked to your secondary antibody was able to stick around because the antigen was there or the other antibody was there that you were looking for. The enzyme that was linked to it cleaves whatever your substrate is. And when it cleaves a substrate, that substrate causes a color change. It's a colorimetric change. It is measurable. How dark it is can correlate with how much antibody there was in there, for example. Rapid testing, just be aware of the general concept of it. It's the same exact principles as this. It's just done on what we call immunochromatographic paper. And so instead of it happening in the well or mixing things together gradually, there's a piece of paper that's like filter paper, and the liquid runs across the paper. And if it hits the line will cleave whatever is going on in there and cause the color change to happen. And it's essentially what's going on there. It's a very watered down version of it, but it's the exact same concept. It's just flowing on a laminar flow filter paper. And when it hits the line, if there's a color change, then that means that the same reaction happened. That's how rapid tests work. All right, the DNA stuff, transformation, PCR, and gel electrophoresis. Yeah? All right. What does PCR do? I know some of you guys were very eager to tell me what PCR stands for. I'm not saying I'm never going to test you on what PCR stands for. We could always save that for the final exam, though. So PCR amplifies a DNA sample. So PCR stands for polymerase chain reaction. It's polymerase. The polymerase being used is DNA polymerase, and we are literally just copying DNA. It's exactly like replication, except we don't have to worry about a huge piece of DNA where we have a replication fork and all of this. We don't need to terminate it because we're only letting it run for a certain amount of time, and however far it got along is as far as it needs to go. But this is basically a method of amplifying DNA samples. What's another way of wording amplifies DNA samples? Making copies. And so every round of PCR that you do, you have doubled whatever you had the previous round. So if you go through one round of PCR, if I tell you you start with one DNA and you go through one round of PCR, how many DNAs do you have at the end? Two. If you go through two rounds of PCR and you started with one, well, if your first round went from one to two, your second round started with two. How many do you have at the end? Four. So you're going to double it every time, not add one on. Literally double it every single time. Whatever the previous one is, double it for the next round. And so that's how we are making copies. It is exponential. It is 2 to the power of X rounds. That's how you would literally calculate it. On the exam, I'm going to expect you to tell me how many DNA molecules you'll have. It'll always start with one, and you need to be able to answer up to 6 rounds. Okay? Should be able to walk it out, but it literally just doubles every single round. All right. Which DNA fragment is the largest from this choice? You will have a question on the test like this one. And I will space out. It won't just be 4 kbp, it'll be 4 space kbp. So it should be easier to see the Ks. So this one will be 35 kbp. This is 35,000 base pairs. So blue is our largest one. Yes. All right, which of the following DNA fragments would travel the farthest or the fastest on an agarose gel in gel electrophoresis? Or if we are running DNA out on a DNA agarose gel, basically DNA it is separating throughout this gel, and that's agarose is literally what makes it solidify, just like the agar in our plates, but it's just a purified version of what makes it solid. And so we've got this gel, and we put DNA in these little wells that we form in it, and then we run electricity through it. And the DNA will run to the positive because DNA is negatively charged. So because we got all those phosphates in the backbone of it, right? So sugar phosphate, so many phosphates. It's so many phosphates. So phosphates are very negatively charged. They're P with 4 oxygens around it. And we already know oxygens like to hog an electron. So very electronegative there. DNA will run to the positive end. But the bigger you are in as a DNA molecule, the more likely you are to get trapped in the meshwork of the gel. So the smaller pieces of DNA travel the fastest or the farthest, and the larger pieces get stuck and they don't go as far. And then we can actually separate out DNA fragments based on that. Very useful in forensics, basically telling the difference between people. Since we all have different genetic codes, we can cut your DNA into parts and then look at the patterns that develop when we run them out on the gel, and everybody's is different. And so the one on these choices, the one that goes the farthest will be the smallest one. In this case, from these choices, that'll be the 100 base pair, which is the smallest. Alrighty, what type of cells are capable of taking up exogenous DNA? What's the best answer for this question, right? So, cells that are able to take up exogenous DNA. Exogenous means outside. So that means you're a cell just hanging out in wherever you are. And somewhere outside of you, there's some DNA. If you can take it into your cell and even express those genes that you took in, then you are called competent. All right. Can pathogens be competent? Some of them can be competent, but there are plenty of other organisms that are not pathogens that can still be competent. In fact, the E. coli that we used as our reference for our thing, yeah, we used plastic Tupperwares in our simulation, but what's commonly used, that was to represent an E. coli cell. And E. coli is commonly used for this process. But these are very well-known versions of E. coli that are non-pathogenic. So they're not even pathogens. And so they're competent. And so we commonly use E. coli. You can use yeast even though that's not bacteria. Yeast are eukaryotes, right? But you can use them. Yeast can be competent. So yes, so any cell that can pick up outside DNA, and not all cells can do that. Competent ones can. So this is dealing with transformation, right? That transformation is the process of a competent cell picking up outside DNA. So yeah, that is a method of horizontal gene transfer. All right, know your DNA fragment sizes. Know that they are documented in base pairs and no kilobases and megabases. Agarose gel phoresis, agarose gel electrophoresis principles. We went over that a little bit. DNA is negatively charged. It's going to move to the positive end. The smaller pieces are able to do that quicker. And then we can separate out DNA fragments that way. how PCR works in a practical sense. So we should refresh our memories on the steps of PCR. The major steps of PCR were denature, using heat to separate the DNA. The second step was anneal. Anneal means sticking together. And so what we're doing is sticking the primers onto the DNA strands. So priming, annealing is a priming step with the primers. And then elongation, adding on nucleotides that are complementary from that primer, building off of that. You cannot add nucleotides without building off of the primer. You can't. DNA polymerase requires that a primer be there. And so if the primer doesn't bind, the nucleotides can't add on, and therefore you would be negative for whatever you're doing the PCR test for. Only if you're positive for it will the primers bind, and then the nucleotides build off of the primers. Um, Bacterial transformation. So I think I remember somebody, I don't know if this was discussing about my class or discussing about something else, but I heard some people talking about like buffers and things like that that were used in PCR. Basics for that, I mean, if you're running PCR, you need DNA polymerase. And it has to be heat resistant because you're going to heat it up to 92 degrees Celsius, which is almost boiling. And most DNA polymerase won't withstand that. Your DNA polymerase would not withstand that. That's too hot. It would denature your DNA polymerase. Not just your DNA, but your DNA polymerase would be denatured. And so we use a special DNA polymerase called TAQ polymerase, TAQ. And it stands for TAQ, comes from Thermus aquaticus, which is a type of bacteria that lives in those thermal vents deep down in the bottom of the ocean. And so those release above boiling temperature. And so the bacteria has to live in those extreme temperatures. And so their DNA polymerase has to withstand those extreme temperatures. We took that DNA polymerase and we use it in PCR. It's revolutionary, basically. It's the only reason why we have PCR is by discovering that DNA polymerase because it's resistant to the heat. So we can separate based on heat, the denature step. We can anneal based on primers that are designed specifically for whatever gene you're looking for. You have to know the gene sequence and you have to be able to design the primers and be able to order them and have them on hand and whatever. So you have to know that stuff about your organism that you are PCRing, okay? So you have to have your DNA polymerase. And DNA polymerase, ACE, always hints at this is a protein enzyme. So DNA polymerase is a protein enzyme, and like many proteins that we learned about in Unit 2, when we learned about conjugated proteins, you guys remember about this? When we talked about cofactors and things like this? Okay, DNA polymerase requires a cofactor. So that means the DNA polymerase itself, the protein portion, is the apoenzyme and it requires to bind to a cofactor, which happens to be magnesium. So magnesium is a metallic ion that binds with the DNA polymerase as a cofactor to create the holoenzyme that's then able to add on the different nucleotides and everything. Can't do that without the magnesium. So we have to have DNA polymerase. We have to have its cofactor magnesium. We should have a buffer in there just to maintain the pH and maintain basically the stability of the whole process. The buffer also contains the nucleotides, the A, T, C, G, that we're going to be adding on. All right. And there's other factors in there as well, just to keep everything stable and have all the cofactors that everything is needing and whatever. So, right. That's the gist of what would be in your buffer. Your DNA polymerase, magnesium, buffer stuff. Why do you have buffers? It's usually to maintain pH and the stability of proteins and stuff like that. And the nucleotides. This is generally what you will have in your buffer. Right. Okay. So that's, and then the elongation step. We already went over that. Yes. Okay. So that's the gist of how PCR works. It just adds on nucleotides and keeps doing it until you stop adding, stop it. Like if that runs at 72 degrees Celsius. So once you heat it back up again to 92 degrees, you're separating the strands again. You stop elongating. There's no real termination step. You just keep going for, 5 minutes and then you separate it again. So there's no real termination step in PCR. You just keep doing it over and over and over again. Okay. Bacterial transformation. So the three types of horizontal gene transfer were conjugation, where we had the sex pilus, which is the channel that forms between two bacteria. And they can shuttle DNA through the little channel. And usually gram negatives are the ones that can do that. We also had transduction. Transduction was the term for whenever a phage can transfer genetic info from its original host carry one of the genes with it and put it into its next host. That transfer of genetic information because of the phage is called transduction, and that's usually a result of lysogenic conversion. It's usually done through transduction. Then we have what we did in lab with our stimulation, which was transformation. That's when the competent cells can pick up the outside stuff. We didn't have any questions about it on here, but you do need to know what gene products are made on each type of media for the exam. There will be a question about that. If the media contains only ampicillin, what gene products are made? Not none. We're still making the gene product for ampicillin resistance, right? So I know we went over that in class, but I just want to touch on that again. If you have media that has ampicillin and arabinose sugar, then you're going to make ampicillin resistance, you're going to make GFP, and you're going to make the arabinose breakdown genes. Yes. Plasmids, what are they? Plasmids, that's what we simulated that we put inside of our E. coli, right? The whole opening it up from the heat, mixing it together with the plasmid gets inside of the cell, and we sealed it in with the cold. That plasmid is a circular bit of DNA. So plasmids are DNA. And so they have genes on them that the cell will read and make the products for the ampicillin resistance, for breaking down arabinose, and for GFP. But the plasmid itself is DNA, and it can be DNA with coding for each of those gene products. Any questions about the DNA stuff? I didn't take a stop for ELISA. Any questions about ELISA or the DNA stuff before we move on? Okay. Eukaryotes. Focus on the differences, please. Focus on the differences, okay? All right, review your drawings, review your pictures. If you took pictures, I have the review sheet also that's online, like on those those lab sheets. On my lab sheets for the eukaryote thing, I believe I have a pictures of the, hold on, I'm saying it like I can just look at it. Lab midterm study sheets. And when it catches up. Okay. I got all the pictures. Now, not all of these are the same as ours. And I usually put a little note over here. Well, our aspergillus wasn't blue. Ours was greenish colored, right? But their penicillium looked just like this. And if you zoom in, you can see the pictures better. You can see, of course, when you're looking at it in the microscope, you can see that the aspergillus had those round dandelion shapes, and the penicillium had this sort of flat, it looked like paintbrushes to me. What are these structures called? It says it here. Conidiophores. Not hitting it again. Okay. Look at this organism transmission route. We should have filled these things out. And I mean, there's more than just these on that sheet, right? But you should have actually gone through. Now it says, who cares, not you, for these. I did not update that from past semesters. You do, because I showed it to you and put it on a microscope and showed you, had it pointed out and told you what it looked like. You do need to know Giardia and Entamoeba histolytica. You need to know it. I know it doesn't say it on this because I didn't update it from last semester. But none of my students could find it, and I got fed up with it, so I just told them I didn't need to know it. But I found it for you guys, both of these. And so to me, whenever you look at Giardia, which was that teardrop shape, he looked like he had eyes, little horseshoe crab guy that was gray in color. That was Giardia. Entamoeba histolytica, he looked like a blob, a dark blob with dark blobs inside. Okay, that's completely different from this purple guy, which they had little purple tails, and they were all over the place. They looked just like this on our slide, versus this guy down here, Trypanosoma. Ours were a little more purple in color, but they look like little snake guys. All four of these look drastically different from one another. I do expect you to know all four of these, okay? Over here, complete the following table. You didn't need to know the group, but you do need to know the transmission routes and their definitive hosts. That was part of the worksheet. And I will absolutely be asking you a question about how does dipylidium, how do you get infected with dipylidium? Apparently by swallowing infected fleas. So if this isn't a hint to you that this is more likely going to be an animal disease, like dogs get dipylidium, that this is more commonly going to be seen in cats and dogs, but they bite at their itchy spots and they ingest the flea and then they get the worm. Okay. Anyways, then the eggs. Ours were slightly different than this. Which one was different was, it depended on which Clonorchus you looked at. And there were two different versions. There was the whole worm mount, or there was the cross section. Hint, hint, Clonorchus is not going to be the one that I put on the slide because there were two different ones and I just felt like that's confusing. But I absolutely will put one of these guys on there. Our Dipylidium one, it was slightly different. Our Dipylidium was pink. It was bright pink. It looked like grapes. Another hint for you guys. I might put a slide up there and just say, what organism is this? Or what organism is this from? Or something like that. And I'm not even going to tell you it's bacteria or anything. If it's on 10x lens and not on the 100x with the oil, if it's on 10x and you're seeing it and it looks like this on a 10x, that ain't a bacteria. Bacteria, we always had to use the 100x with the oil. So we had to have 1,000 times magnification in order to see our bacteria. If you're not understanding where I got 100x and that became 1,000 times, then you should look into that as well, because you do need to know that for the exam as well, calculating the magnification. Your eyepiece, where your virtual image is being made, the eyepiece is 10x. The light travels up through the specimen into the first of the objective lenses. We rotate our objective lens on the nose piece, right? The nose piece is the part that rotates. The lens itself is the objective lens. And so we have the 4x, 10x, 40x, and 100x. Every time you use the 100, you always use immersion oil to reduce light scatter, right? So, but that, whatever lens you're using, let's say it's the 10x, times your ocular lens, which is always 10x. And it says it on the lens too, in case you forget, you can look at it. So you just multiply those. So 10x times 10x would be 100x. But if you're on the 100x lens, then that is 1,000 overall because you have the 10x times that. Okay. So that's how we get to that. But if you're looking, it will entirely be multiple choice, entirely multiple choice for what it's worth. Yes. So if you're looking at a microscope and I'm asking you what the total magnification is, then yes, you will need to know how to calculate that. But yeah, if your microscope is set up at 10x and it looks like this, you can clearly see like the pink grape-like structures. Y'all, that ain't a bacteria. It's unlikely it's even a protozoa. Because even these guys, I usually had to go at least 40x to get these images. So like probably a helminth, and we only looked at the eggs for the helminths. So I'm not going to add it. When I ask you a question about your helminth eggs, it's not going to be Clonorchus because we didn't all see the same thing. It will be one of the other ones. Focus on the differences. Okay. Any questions about the eukaryote stuff? Okay, we're going to have some questions about them. Which organism is this one? Alrighty, this was Penicillium. Penicillium. And what type of organism is Penicillium? It's a fungus. It is in fact even more than just fungus, it's a mold. And that structure that we saw in this image here, this is a conidiophore. What is the name of this organism? All right, so what you were seeing there was trypanosoma. It is the cause of African sleeping sickness if it's Trypanosoma gambiense, like what we saw in our slides in lab. There's another trypanosoma That is Trypanosoma cruzi, and Trypanosoma cruzi causes Chagas disease, which if you donate blood, you've probably heard about this, but it is a bloodborne disease that is caused by this protozoa. This is a protozoa, and all protozoa are single-celled eukaryotes, always single cell. They're never multicellular. I know that it looks like it's got all these parts going on in it, but that's just one cell. That's Trypanosoma. All right. Know the organisms that you either had to draw or that you took pictures of or that I have posted on that study sheet, right? And be able to tell the differences between them. And we had the sheets that we had to fill out on our eukaryote worksheet. Be sure you actually have gone through those and answered the questions associated with them so you know the major differences between those guys conceptually as well. Know how they're transmitted. That was part of that sheet. For sure, how they're transmitted. Identify them based on their major differences. I'm not going to ask you what their major differences are. Just you can look in one and see that it's purple grapes, then that's going to be dipylidium. If you look into one and it has that weird sharp thing on it, the schistosoma, it has this sharp thing on sticking out the edge of it. So focus on differences like that to help you with defining, like remembering which one is which. Understand what the term trophozoite and cyst are, because that was part of our eukaryote lab as well, where the trophozoite is the active free living form. It's equivalent to the vegetative bacteria. The cyst is very similar to the endospores of bacteria. Now, endospore only applies to bacteria. Cyst only applies to protozoa. And but otherwise, other than that, they're very similar concepts. All right, bacteria, general bacteria inoculation, growth, isolation, and things like that. Which of the following is not a term used to describe colony morphology? Colony morphology. So remember, colony morphology is what we saw with our naked eye growing on our plates. And so one of these doesn't really have to do with the colony morphology. This one's a tricky one. All right, so on your exam, the lab midterm exam, you will never have more than 5 choices on the midterm. Anyways, the final, you might have more, but on the lab midterm, you will never have more than 5 choices, just so we're clear. You also won't be able to choose more than one. One answer will be correct, okay? But one of those answers might be all of the above or none of the above or whatever. So keep that in mind. But anyways, so this one was hyphae. So hyphae is something that you see at the microscopic level for your molds. And so it's like you take a mold and you look at it under a microscope, if it's branching off of the other ones and creating all these branches, those cells kind of branching off of one another, those are the hyphae. So rhizoid, punctiform, plateau, irregular, and convex are all terms for colony morphology. I'll give you a really big hint for what type of questions I'm going to ask on the test for colony morphology. It's going to be just like this. It's going to be, which of the following is a colony morphology term? I'm not even going to ask you to define the term. It won't be one of these. Okay? I'm just saying, it's not going to be one of these. But one of the other terms, and it's not going to be mucoid, like an obvious one. So there's some of the terms where if you go to your colony morphology stuff, let's go back to it. It's exercise 2, where we've got these terms here. Raised. It's not going to be raised. Umbonate. That's a fried egg shape. Flat, convex, pulvinate. It's going to be one of these kinds of terms is what I'm going to say. It's going to be one of these ones where you're not just going to convex. Most people know what that means. It's not going to be that. It's not going to be matte. It's not going to be mucoid. It's going to be one of these other terms. Okay. Know that it is a colony morphology term. I'm not going to ask you what it means. You're welcome. All righty. Next question. What is the name of this type of cell of culture? This type of culture here. So this is the tube upright. If it was sitting in a rack, it would still look like this. I would say there's a hint in the image. A little bit of what this could technically be. And that we use these all the time in our lab. This is the slant. So this is a solid media that is at a slant inside the tube. It's not in liquid form, right? So we know the plates are solid form, the auger plates that we use, solid form. The slants, they are auger, but at a slant. A butt is auger, but it's not at a slant. It's just, it looks like, I'll show you what it looks like. It looks like this. The tube with your little lid on it. This is not meant to look, it's going to always, no matter what I do, it always looks weird. And then on the inside, it's just flat and it's like all filled in. Okay. This is like the auger part. And this, just pretend like I filled this all in really good and it looks great, right? Okay. That's a butt. So butt is just flat. It's like, it looks like a broth, but if you tip it, it's solid. That's a butt. So plate, slant, and butt are all solid media, and they're solid thanks to stuff that we add to it that makes it solid, which was auger, which we went over like way back when. That was like exercise 1 or something. So, but you do need to know that. And which one is liquid? Broth. It's the only one. If it's liquid, it's a broth. Okay? All right. Oh no, undo that. Close that. There we go. Okay. All right. How was this organism isolated? By what method did we isolate this organism? We are almost done, folks. We'll be done just a little bit. Just want to be thorough. Alrighty, so this is the streak plate. We also call it the quad streak plate because there's 4 different quadrants to it, right? It's just a way of taking a sample, putting it on the plate, fire your loop, right? And then you go in again with your sterile loop and spread it out. The whole point is that in each quadrant that you do that with, you're just spreading out the cells more and more. It's a way of diluting it quite physically without actually diluting it with liquid. Just spreading out those cells so we can get individual cells, which will grow into individual colonies, and then you can create pure cultures from that. Which arrangement or arrangements of bacteria do you observe in this image? Be careful and focus on what the question's asking. Arrangements, groupings. All right, this was not cell shape, not cell shape, arrangements and groupings. So I just want to reiterate that. So on the exam, be sure that you are looking at what I'm asking about. So if I'm asking about the arrangements or the groupings, I'm not asking about cell shape. If I'm asking about cell shape, that'll be in the next slide. The next question, by the way. So this one was just strepto. Now I could see how some people could kind of parse out staphylo, but the only reason that we would even get to that is this area right here. But that's just the chain kind of folding back on itself. Do we really see true like clustering of these guys anywhere really on here? I would not say so. Something you could say you saw on here that wasn't on one of your choices, I took it out on purpose so you didn't get confused, was diplo, because we have diplo right here. But overall, this is strepto, which is the chains. Okay. What is the name of the shape of this bacteria? Alrighty, so that's bacilli, the rod shape. You only have 2 choices. I mean, only 2 shapes that could possibly appear, which would be either bacilli or cocci. I won't have any other, I don't have other bacteria that do the other shapes. There are other shapes, but the only ones that I have are either cocci or bacilli. So it's going to be one or the other. All right, know your colony morphology terms. Again, I won't necessarily ask you to define them, but know that it is a colony morphology term, okay? The types of media, know which ones are liquid or which ones are solid. The isolation techniques really just focus on the quad streak method, okay? And which quadrant's going to have the most single isolated colonies would be the fourth quadrant, right? Cell shapes, cocci or bacilli. Cell arrangements, focus on strepto, staphylo, or no arrangement, because I don't have bacteria that do anything else. Okay. All right, microscopes and how to use them properly. If you observe a specimen using your 40x objective lens and have 10x ocular lens, what is the total magnification? Very common question on my test. Exactly, you multiply it, you get 400. Beautiful. Which objective lens do we use for oil immersion? In our microscopes, which one? Yep, that's the 100x. 100x is oil immersion, and you always use the oil with it. All right, be able to calculate magnification. We've gone over that. Understand how the microscope operates. Generally speaking, where the light comes from, the light source, which is down here, then it goes through the condenser here. You can open and close that aperture, which we call the iris. You can open and close that using that switch that we call the diaphragm. This is the stage where your slide sits on top of. The slide sits in here between those stage clips. The light travels up through from that light source through the condenser and the iris and the diaphragm through the slide and then hits these things here. These are called objective lenses. The piece that the objective lenses are rotated with is called the nose piece. That black piece up there is called the nose piece. That black piece is not the objective lens. These things that hang down, each of these is an objective lens. The light hits the objective lens and creates the real image. It's the first time that we're really creating an image. There's some mirrors in here that then reflect it out to this piece up here, which is your eyepiece. There's another lens in here, which is almost always 10x. And so when it goes through that lens, it creates the virtual image because we're changing what our original real image was through another lens. And so real image down here, virtual image up here, whatever your objective lens is down here, 10x, let's say, and then your eyepiece up here is always 10x. So you multiply those two, that would be 100 overall, or whatever your objective lens is on. So know your parts, know what each of the parts does and how to use them, and understand why we use immersion oil. Remember that the light comes up through the glass, and when it hits the air, that's a drastically different density of substance. So when it hits the oil has a more similar density to the glass, and so that prevents light refraction or light scattering. So that's the whole point of that oil immersion, not to lubricate the lens. Okay, Kirby-Bauer, which was our antibiotic susceptibility and resistance test. Remember, the bigger your zone of inhibition, the bigger it is, the more susceptible. That means that the antibiotic, when we look at this, right? Let's look at whatever this is, LZO. I don't know what this is, but let's look at this one down here. This one has the biggest space around it, right? And so this one down here, we would say that this is the antibiotic that this particular bacterial species is the most susceptible to. Which one would you say it's most resistant to? I would say this one, whatever that is, and this one, which I think is azithromycin. I don't know what this one is. And then we have CIP, which has, it's not a good choice. This is Cipro. This would not be a good choice for this bacteria. So we would go with whatever this LZO is. Right. So, that's how you would kind of look at that. So this area where there's no growth, we call that the zone of inhibition. And we could actually use this zone of inhibition, whatever we measure it to be, to calculate out what we want our minimum inhibitory concentration, MIC, of our antibiotic to be. And so if you've gotten to chapter 12, then you're definitely learning about the minimum inhibitory concentration. If you haven't, that's okay. I know that's not until technically I'm not, it's whatever. We have this to worry about now, right? So, but that minimum inhibitory concentration is calculated using your zone of inhibition. That's the minimum amount of the antibiotic that you would need for it to be effective. And so you don't want to use too much more than what you really need, because then that could lean into aiding and favoring antibiotic resistance. So, and then yes, so zone of inhibition, you measure it all the way across the whole diameter across in millimeters. The larger the zone of inhibition, the more you inhibited, even at lower concentrations. So you get lower and lower and lower and lower the further away you go. Is there still, if we look at CIP, is there still CIP, ciprofloxacin antibiotic out here? Yeah. Yep. Yep. Yes. It's just, this is the only where it is even remotely effective. And it's not very effective for this guy. So yeah, so that's the idea. There's still CRO antibiotic even out here where there's growth. It's just you need at least this much of a concentration for it to inhibit the growth. Okay, so the further out you get, the less of that antibiotic there is. And so the closer you are, the more of that antibiotic it takes to be inhibiting growth. Do we know if this LZO, do we know if it killed it? We don't know yet, right? We know it inhibited the growth here, but we don't know if it inhibited it because it killed it or if it just inhibited the growth. And so remember, we would sample in this clear area and put it on a new plate. And then if it grew, it just inhibited it. If it did not grow, that it killed it because it was all dead here when we tested it. And so that's where that cidal versus the static comes from. If it's cidal, it killed it in here. If it's static, it just inhibited. But you don't know that from looking at this plate. You have to sample in here, grow it on a second plate, and then see if it grew or not. We tracking? Do you guys have any questions about that? Because there was some confusion about that on that lab exam or lab worksheet. Any questions about that? All right, here's our question. Which antibiotic is the bacterium least susceptible to? All right, excellent. We all chose E because there was literally, it just overgrew it. Whatever that antibiotic is, it doesn't affect this guy at all. This guy is completely resistant to E. So if it's completely resistant, it is not susceptible at all. So that's the idea with that. All right, know what zone of inhibition is, have an idea about minimum inhibitory concentration and what that represents. We have Mueller-Hinton auger is the auger that we use. Why do we use it? Because it's pH controlled, it's depth controlled. Why are those important? We want the pH controlled to help with the stability and the functionality of that antibiotic. And we want the depth controlled so we can have that diffuse evenly throughout the plates, not just that plate, but all plates that we've made so that we can compare the results to one another from plate to plate. So that's why we use Mueller-Hinton auger. It's just a more controlled environment to control that diffusion of that antibiotic as well as the stability of the antibiotic. And know the difference between bactericidal and bacteriostatic and the ability of how you would determine that from the assay or steps that follow this particular assay. So this is the Kirby-Bauer assay where we have the actual zone of inhibition. The next step that we did after that is where we determine if it's bactericidal or bacteriostatic. Unless you do that second step, you can't tell from the Kirby-Bauer assay. All right. All right. If you have any questions about any of this, please don't hesitate to contact me. Happy studying. You guys know that I'm pretty communicative. And let me know if you have any questions. I am here on campus until 12:30. I'm just going to be setting up the exam in the lab. So if you happen to be on campus and you have questions and you want to come see me, I will be in the lab. So I'm just setting up the exam. So if you do come by, we'll have to leave. Like I can't let you see the exam. But if you are on campus and you want to come see me in person, just come by and we'll step out into the hallway and talk about whatever you want to talk to, or go to my office and talk about whatever you need to. But I'm only here till 12:30. But you guys can email me anytime. You know how that goes with any questions that you have. Here's our results and our little podium. Anybody have any questions for me right now while I'm here? Now is the time. You guys did great. I feel like we covered everything pretty well. And like I said, feel free to email me if you have any questions. That's the best way to get in contact with me would be through email. I check it pretty regularly. Otherwise, good luck. I'll see you guys tomorrow at 10 a.m. We will rotate. Like I've said, it will have 1 minute per station. It says 1 minute per seat. And it seems like it's not a lot of time, but I promise you it's more time than you think. And you have plenty of time after we have gone through, there's only 24 stations. So it's 24 minutes of rotating. We have all of the rest of the time and then some, because I don't have class after that or anything, for you guys to go around and go revisit questions and make sure that you're getting everything. All right. And it'll make more sense when you get into class tomorrow. Be sure you have your lab coats. Be sure you have closed-toed shoes. And be sure that you keep your phone silent, do not disturb, in your pocket in case you need it for anything. I will provide scratch paper, just a little same scratch paper if you want it. You are allowed to use scrap paper. You are allowed to dump, and we'll go over the details on how the setup works and how to move and everything on the test day. I promise there's plenty of time. There's no rush. We can take our time with everything and make sure everybody feels comfortable with it when we get there. Any last second questions? All right. If that is it, good luck, happy studying, and email me with questions. Have a good day, everybody.